So on high level, what we are doing in Rubedo is really to identifying and discovering the pathological cells that emerge with the aging process. And we do this using a computational platform that I'll present. And then once we do that, translating this into small molecules using a chemistry platform, small molecules that are engineered to target those cells selectively, taking advantage of metabolic activities. And we're really focused right now on chronic, progressive, inflammatory, and fibrotic degenerative diseases of aging. With a team, cross-functional team across, drug hunters from big pharma, biotech, experts, KOLs in aging field, and Silicon Valley tech experts. So I'm going to talk a little bit about today. Should set the timer here. About a little bit of the heterogeneity of senescent or senescent-like cells. They're not the same, and so which target should we go after, and which tissue and how? And then how we use this to really translate into therapeutics using our approach and computationally how to find those cells and identify this heterogeneity and how to translate this into therapeutics. So we're very familiar with that, right? The aging is the ultimate factor of most of the disease of aging. But we already talk here is obviously the biology of aging, not the chronological aging. And I think what is really intriguing is this is a very plastic phenomenon. So this inflection point here can actually be changed. And it actually can prematurely accelerate it. There are many cases. Cancer survivors experience premature aging. They experience age-related diseases, so this inflection point decades sooner. Same for severe cases of COVID-19 survivors. So this true also can go in the opposite direction. But so what's happening during this inflection point? There is a change in biology, in physiology, in metabolism. And there are an emergence of cell types that are really pathologically driving this process and diseases. And most obvious known example is cancer cells, but there are many others. Really, some are even unknown. And one example that is better known, although not fully understood, are senescent cells. And we rather call them senescent-like cells. So what are those cells? The senescent cells, and I think most of the people here are familiar with that, are cells that any cells in the body can be triggered into this state by damage and stress. They stop dividing. They become very resistant to cell death. And metabolically very active, they secrete a number of factors, the secretome, that has the function to really call the immune cells to sight, to recruit in this immunosurveillance. And this is good. They can be cleared and improve and promote tissue regeneration. Those cells could be the results of tumorogenic stimuli, and they can be potential cancerogenic cells. So there's a tumor suppressor mechanism, and you need to get rid of them. But they have a pleiotropic function, so they can also play the opposite role. And especially with aging, persistent damage, and disease, they can, for different reasons, escape the immune cells and actually trigger that to participate in driving now a chronic inflammatory process that is pathological and results in tissue degeneration, fibrosis, and stem cells depletion, or even promoting cancer. So, really playing the opposite role. So they've been identified really to be behind many age-related diseases. Now, importantly, they're not just pleiotropic, but they're also very heterogeneous. So they're not all the same. As a matter of fact, there is no such a thing as a senescent cells. It's like there is not a cancer cell. There are many ways to trigger and induce a cells to become senescent. They've been identified in vitro and in vivo. But what we and others found really that a cell can move into different state of senescence, so that are differently regulated, and they can play different roles in different tissues and disease. So I'll give you an example of one of those modalities, like cytotoxic drugs can induce cells to move into the senescent state. Now, what has been shown, and we know, that cancer survivors, even young people that sustain standard of care in cancer, they prematurely age permanently in many cases. Their quality of life is decreased, and they really, even young pediatric cancer survivors, for example, they are characterized by an increased expression of markers associated with cellular senescence, like P16, but also physiologically and functionally, frailty scores appear to be increased in these patient populations. Sarcopenia. And we're talking about 20, 30 years sooner compared to the average person, which that happens after 65. So, for example, what we did work around to understand a little bit better this process. And so if we look here, this is AMR90 is a human-like fibroblast cell line. Then we induced senescent cells with the same modality. So it's using, but two different standard of cares. In this case, it's doxorubicin or doxorubicin plus fluorouracil, which is typically used in treatment for breast cancer and others. Now, looking at traditional markers of senescence, like P16 or beta-gal and others, those cells look the same. They move into a senescent state. So you would expect that to respond to senolytics in a different way, and this is not the case. If we use that's a first-class senolytics, like navitoclax, which are repurposed drugs for the cancer space as BCL2 class inhibitors, we see that in one case it's with a very good and potent senolytics, but in the other case, you start to lose potency and selectivity. So we characterize that. If you look at gene expression, we see that there is very little overlap. So between upregulated and downregulated genes, suggesting that really they're moving into a different transcriptomic profile landscape. And more sophisticated pathways analysis shows that this is exactly the case. Probably a bit small to read, but like extracellular matrix remodeling pathways can go really in the opposite directions. This is the same cells. They are senescent cells, but they've been triggered in this state using two different chemotherapeutic agents. So this is actually more important if you move from a cell line to primary cells. So now we're looking at patient-derived, lung-isolated, fresh-isolated cells from IPF patients that have pulmonary progressive fibrosis, where this senolytic doesn't work anymore. And not just in humans, but if you look at rodents, so mice, rats, or hamster, shows that navitoclax doesn't show a senolytic index in these IC50s in vitro, suggesting that in this context, this is probably not the right target. Not saying that navitoclax is probably good senolytics in other contexts. So that's just an example, because so we really, over the years, developed multiple models of senescence and characterized that using the platform of how to identify and profile this senescent state. I'm showing an example. This is a matrix of 10 different cell lineages and 7 methods of inductions. You see that there is a high degrees of heterogeneity and variability. Again, convergence or divergence of those states. And when we compare with, again, patient-derived IPF primary cell from single-cell RNA sequencing, we see that certain models recapitulate this much better and others are completely different. So again, you need to also know which models to use for a drug discovery effort to make sure that you go after the right targets. So to really help elucidating this, we really built the platform around how can we understand this heterogeneity at single-cell level. It's important single-cell level because those cells are rare. They're not all large, they're intersparse, and not just heterogeneous, so they're also maybe sometimes hard to identify. And so we built this platform that is an AI-driven multi-omics based on single-cell technologies that can really triangulate and help understand among those cells which we should go after and how to characterize them. So the platform is, at this point, fairly sophisticated. There's really multiple functions and different algorithms that really work through that. And kudos to our CTO, who actually an outlier in the space, came from Facebook and other tech companies, where he was one of the lead software engineers there. And really works through the user interface now, where our biologists and chemists can go into and interact with the data from an identification of those cells and the targets and the functions associated with that in a bioinformatic part, and then down the line using chemoinformatics for medchem and the molecule designs, etc. And preclinical models developments based on those. So today I'm just showing a little bit of this very first step. And for example, one of the things that we generally start is taking single-cell RNA sequencing as well as other omics data, and ask questions, like in this case, our data from IPF patients, from the lung of IPF patients, first in an unsupervised and then in semi-supervised way to classify what are those aberrant cells that emerge, and among those clusters, which are the ones that seems to be the most significant. So then we can really isolate, characterize, and validate targets experimentally, and do analysis such as pathways analysis, suggesting what are potential targets and the molecules associated with that we can screen against with. But there is another thing that we do, which is in those cell types, what are the metabolic functions that are associated with those cells, like enzymes and other cellular activities that we don't think as the druggable targets, but more the mechanism of action that we need to use to selectively target those cell types. So what does it mean? So I'm going to show an example, and this is based on from one of our early work. It's a proof of concept that we did, where we showed using, in this case, not something that we discovered through the platform, but a known metabolic activity and enzymatic activities. That is beta-galactosidase, is one of the markers associated with cellular senescence, to generate a prodrug derivative that can selectively target that. So what does it mean? That we can, if we identify a target or a molecule that can, in this case, kill senescent cells, or your target cells, and then we can use then metabolites of enzymatic activities that are expressed when the cell is in this state, as promoietives that can be engineered to inactivate this warhead. And so asking your cells to be converted and activated intracellularly. So in this case, using a galactose derivative prodrug of a senotoxic that we screen, and through known agents in this case, in these studies that is published, and we found, in this case, fluorouracil, and we generated a galactose derivative. So I'm skipping the in vitro part, but just going to in vivo. As we know, this parent drug has toxicities, so not surprisingly shows toxicities in this case, young mice, hematotoxicities, loss of body weight, and others. But the prodrug derivative can normalize this back to a cell in control, so preventing these side effects. This is more important in age mice, geriatric mice. We look at 28 to 30 months old mice, so very old, where a single dose of the parent drug results very toxic and is not tolerated. But chronic treatments, multiple treatments for weeks of the prodrug derivative shows this to be well tolerated in these mice, and even at an extensive hematotoxicity profile shows that those cells are this prodrug, senolytic prodrug, is well tolerated. So to study the effect based on this MOA, we generated similarly a prodrug derivative of a luciferin, which is a substrate for luciferase. And we generated geriatric cohorts of mice that expressed these luciferase enzymes so that they can emit lights, but only when this prodrug is activated in cells that are high level of this enzyme. And we see the young mice don't, but the old mice, they glow when they get this prodrug, similarly to young mice that receive chemotherapy, that, as we saw, they acquire this sort of accelerated phenotype of aging that then we validate histologically and by gene expression, looking at established markers like P16 and beta-gal. And this bioluminescence level nicely correlates with functions. We have this functional score where we look at different activities like grip test and balance, etc. So using the models like that, we really quantify the capacity of this prodrug to clear senescent cells, at least by this definition. And I'm skipping this part when we characterize this in multiple tissues, and we focus in two tissues in particular, which is muscle and brain. And so in this case, we really we aged the cohort of genetic models. In this case, it's a PAX7 clear-1FP mouse model, which means cells that express PAX7, that are typically muscle stem cells, drive the expression in inducible way of a fluorescent protein. So we can trace muscle stem cells in these mice. And so we aged them up to 30 months of age, and then we started to treat them for a month, multiple times a week, keeping them in cages that have computerized control, running wheel and other sensors, so we can monitor their activity. We saw that those mice during the treatment, they increased their spontaneous activities, and measuring different parameters, we see that overall their frailty score improved at the end of the study. prior to sacrificing them. And that also came with an improved survival. And again, this is not a longevity study because we just across the course of the study, but definitely shows a significant change in survival of those mice that are really very old. So over these months, many were naturally dying. Now we look at muscle and we did electrophysiology. So we isolated single muscles and we stimulate them and we can measure their force production. We see that these mice at that age have this sarcopenic phenotype. Muscles tends to become atrophic and lose functions, but the ones that receive the treatment shows any significant improvement in force production. When we challenge this muscle tissue by injuring and look at their regenerative capacity, we see shifts towards a more useful profile. We see the regenerating fibers, centrally nucleated myofibers, becomes larger, and there is less fibrosis that I'm not showing here. So then we can isolate the muscle stem cells, and we see there is an increased number and generally activity, self-renewal capacity proliferating. So again, suggesting that by clearing those senescent cells, or at least those cells that have high level of beta-galactosidase systemically, we reduce markers of inflammation, fibrosis, and improve really muscle stem cell functions in this case and muscle functions. The same is true also in the brain. So we look at their cognitive functions. We did behavioral studies like Y-maze, Morris Water maze, training them to learn where a platform is as we swim into a small swimming pool. And typically those geriatric mice, they can't learn. They really, they swim randomly, while a young mouse can go very straight to the platform at the end of the training. But these geriatric mice, they perform as well as young. They're just slower to swim, and they retain this memory over time. So when we look into the brains, at the hippocampus of these mice, we do see that geriatric mice have a significant increase in neuroinflammation. Look at microglia expansion or activation in the microglia in the hippocampus, which is significantly reduced after the treatment with the senolytic prodrug. And we don't see changes in neurons, but we see reduction in senescence markers, P16, P21, gamma-2AX, I'm showing here a couple, and interestingly, an increase in neurostem cell activity, like in muscle, suggesting that this brain recover cognitive functions and neuroplasticity, reduction of inflammation. So those studies really overall suggest that using a metabolic activity prodrug could be a strategy to increase tolerability, selectivity, and once you know what to target. And we did these studies in collaboration with Professor Tom Rando, Stanford, my former mentor, and Tony Vaiscori as well, and many others at Rubedo. So this is one modality. In this case, are these, as I mentioned, inactive prodrugs that take advantage of. This pilot was one enzyme, but we can use multiple enzymatic activities to be activated when the cell's in this specific state without harming the good cells, so avoiding the chemotherapy effect that accelerates aging. So that's the opposite we want. And so this is an example of how a compound and finding a novel target is a potent novel compound that we discover that target intrinsically more senescent cells in red more than proliferating or quiescent. But the prodrug derivative shows a shift in order of magnitude in selectivity, so increasing the therapeutic windows, which is a much better candidate. So we really build a pipeline going after several indications that use different, not necessarily this type of prodrugs, we have different modalities. And these are all entry points in indication for larger therapeutic areas and then really age-related syndromes that we are advancing into clinical stage. So with that, really to summarize, we show that there is a heterogeneous cellular senescence or senescent-like cells or pathological cells in general that needs to be taken into account to identify the right targets in the right context and patient populations. And we leverage multi-omic strategies to identify that and translate into a metabolic activated type of senolytics or other molecules.